Thank you very much for the introduction. My name is Florence Raynaud. I work at the Institute of Cancer Research and at the Royal Marsden Hospital in Sutton. And this is the building that I work in the Center for Cancer Drug Discovery. We actually moved in there last year in the middle of the pandemic. It's a beautiful new building. And we are a drug discovery unit. And in academia and in the institute, we have discovered all these compounds here. So it's not bad in the last 20 years for an academic institution. There are about 180 of us. And all these agents have been tested in the first-in-man study at the Royal Marsden Hospital. And I've been involved in the preclinical and the clinical development of these agents. There is one abiraterone that's made it to approval and actually has revolutionized the life of men with prostate cancer. So actually it makes you get up in the morning and do your job. Drug discovery is a team sport. If you're an academic and you want to work alone, this is not for you. And there's a lot that can go wrong. This whole cycle takes about 10 years. And you've got to first have an idea of something to target. So you've got to identify your target. Then you've got to find a way to screen and get hit matter. So we only work on small molecules, drug discovery, but you need to make sure that you've got the right assay to actually identify compounds. And then you've got to optimize these compounds. And to optimize them, you've got to select some biomarkers. And eventually, you've got to make sure that the compound that you have will be able to get to man. So you have to find the best candidate out of what you have. And you could still get it wrong if you don't design your clinical study properly. So you can see that it's not that simple. So to start, you have to validate your target. And that takes into account a lot of the basic biology that we deal with. And so you've got to make sure that if you knock it out, it has the opposite effect if you knock it back in. So it's a lot of genetic manipulation, but also understanding of the protein network, if your target is a protein. And for that, LC-MS and proteomics is quite useful, but this is not what I do. Then you have whole teams of people who work in biological assay development, and they will screen libraries of compounds, sometimes hundreds of thousands of compounds, and you will get some hits. And these hits are normally not very potent against their target, and you need orthogonal assays to make sure that you can separate the wood from the trees. You also need to test these hits in batteries of assays to optimize your assay, so to optimize your chemical matter. And from that perspective, having a crystal structure is really valuable, but there are other biophysical assays that allow you to actually find the structure-activity relationship of your compounds and improve your chemical matter. So your chemists are continuing to make compounds, and they've got to be optimized to the point that you can understand that in a tumor cell context, you can treat with your compound and see engagement with your target. So you need a target engagement assay. And this results in growth inhibition. So when you've got this growth inhibition, you're quite happy because you've got quite a good compound that works in vitro. But of course, it's not all, because you're then going to want to actually optimize the property of your compound in an in vivo model. And to do that, you need good pharmaceutical properties, and you need properties that you're going to be able to translate to man. And this is when my job starts. Really, what we do is we have a lot of ADME screening methods, completely LC-MS driven, that allow us to understand and predict if a compound is going to be absorbed, distributed, metabolized, and basically what is the driver of clearance for your compound. And these assays, particularly for absorption, they're normally cellular assays that allow you to tell if a compound is permeable or not. For the distribution, you can also use these assays because they can tell you about some transporter-driven elimination. But for the distribution, what we use most is a plasma protein binding assay because the free fraction is the fraction that we utilize as the active fraction and that you can scale up across different assays. So what you're saying is your free fraction in a tissue culture media is the free fraction that will be efficacious in vivo. So we often use equilibrium dialysis. And then, of course, we have to do metabolism. How is a compound eliminated? Is it metabolized? And we have different in vitro assay, microsomes, hepatocytes, S9 fraction. And then it's not only how a compound is cleared, but what is it cleared into? Is the metabolite active? Is it toxic? So you need to be able to identify that. And of course, LCMS is the way we do this. So overall, these ADME screening tools give us an idea ahead of time of what is going to drive the elimination of our compounds and whether we're likely to have a good compound for in vivo testing. And then, of course, you're going to do a PK. So you've got a concentration time profile. And what we're saying is that it's the free fraction which actually should compare to what we've seen in our in vitro assay. Now, should we go further on with this compound or not? And we can start to be doing, simulation. What is it going to look like if we give it twice a day, et cetera, et cetera. So at this stage, we've got an idea of the pharmacokinetic properties of the agent. We're then going to take it to the next level. And really, this is the crux of what we do. We have to try and build a relationship, bearing in mind that these targets are novel, so normally nobody's worked on them, between the compound levels, the PK, the target modulation, the PD, and the efficacy. So what you want is you want to understand how much you need to get the desired effect on your target and how much of that desired effect will result in efficacy in your model. So in the preclinical study, of course, we've got mice. They bear normally tumors, sometimes human tumors, so you can sample a tumor quite regularly. That's fairly easy. And you can have your biomarker in those tumors. And you can build your golden triangle and you can understand your relationship. Now, when we go to first-in-man in oncology, first-in-man in oncology are not healthy volunteers, unlike other therapeutic areas. Our phase ones are cancer patients. They normally have no therapeutic hope, so they've gone through all phases of therapy, but they have normal liver function or kidney function such that the disposition of the compound is not affected. So in the clinic, we're going to do essentially the same thing. We're going to actually look at the compound levels, and we're going to check that they modulate the target. And if they do, then we'll enrich at the dose, because that is phase one is a dose-ranging study, and we're going to check that we've got our metrics right. So in PK, we've gotten quite good over the years at predicting PK. Never got it wrong so far, so maybe I shouldn't say that, I'll jinx it. But so basically, we are able to predict human PK from our preclinical data. Now, with the PD, it's a bit more tricky because here you've got animals who bear tumors. You can sample your tumor at regular intervals. Now, in the cancer patients, to start, not all patients have got accessible tumors. And for those who do have accessible tumors, you're lucky if you get a pre-treatment and a post-treatment biopsy. So you're not going to get anywhere with this. So you kind of need biomarker maybe in surrogate tissue if you get expression in those tissues, or circulating biomarkers. And so in addition to the PK, what I've done is, in my career, is really found biomarker metabolites as support of drug development. And we've actually shown that you can really do this, use circulating metabolites, endogenous metabolites, to actually drive drug discovery projects. So what we've done is we've established metabolomic signature in preclinical models, so in those mice bearing tumors during these therapy studies. And what I'm showing you here is 3 different signal transduction inhibitors where we've been able to define metabolic signature in those animals that we've been able to recapitulate in the clinic. So these were a PI3 kinase inhibitor, a MEK inhibitor, and a ROCK-AKT inhibitor here. So the first compound, we had an on-target insulin-resistant phenotype. The second compound, the MEK inhibitor, we had a metabolic signature of response. So those patients who responded had a target engagement metabolic signature that we could see, and we could even predict response with a pre-treatment lipid signature. And the third compound, the ROCK and the AKT inhibitor, we could show that the metabolic signature was linked to nitric oxide synthase, and this was actually showing in paralleling the hypertension that was dose limiting in the clinical study. And of course, all these signatures are established with LCMS. So overall, drug discovery is really a team sport, and it's really supported by LCMS throughout for understanding the biology. Proteomics is essential, and it actually helps us define some biomarkers, proteins, that are being used throughout drug discovery and development. But certainly mass spectrometry of small molecules, especially small molecules inhibitors, actually helps you optimize ADME, metabolite identification, which can also help you choose the toxicology species. And metabolomics really helps you support biomarkers. And the pharmacokinetics in first-in-man studies is obviously done with LCMS. So with that, I'll conclude, and I'll thank you for your attention.